Good morning, everybody, and welcome to this webinar hosted by the University of KwaZulu-Natal College of Health Sciences. Today, we are unpacking the SARS CoV-2 virus and its variants that we obviously know have emerged over this very short time that we had COVID-19 with us. With me, I have a panel of two other panel members who will be joining in this discussion. My name is Kanye Msomi. I'm a clinical virologist by training, and I am based at the University of KwaZulu-Natal in the School of Laboratory Medicine and Medical Sciences, the discipline of virology. I am also the head of department for the National Health Laboratory Service, which offers a diagnostic health service to the public health sector. I will now introduce my other panel member, who is Prof. Tulio de Oliveira, who probably needs no introduction. Prof. Tulio is a bioinformatician. He received his BSc from the Federal University of Rio Grande do Sul in Brazil, and later finished his MSc and PhD in the University of KwaZulu-Natal here in South Africa. He is a well-renowned scientist with a lot of accolades under his belt. He's received two prestigious fellowships from the European Commission, as well from the University of Oxford from 2004 to 2006. He studied at the Wellcome Trust Institute in Cambridge and the University of Edinburgh, studying bioinformatics and genomics. And he currently heads and is the director of CRISP, which is the KwaZulu-Natal Research Institute and Sequencing Platform. He is a well-researched and well-published author who has published in many high-impact journals, top scientific journals, including Nature, Science, and The Lancet. Welcome, Professor Tulio, and maybe if you can just say your hellos.
Yeah, thank you, Kanye, and thank you, UKZN, to organize that. Yeah, of course, it's a great pleasure to be talking with our fellow colleagues, and I hope we're going to have a very exciting discussion. Yeah. Mm-hmm.
Thank you. And with us also, we have Dr. Richard Lascelles. Dr. Richard is an infectious disease specialist. He is also based at CRISP, which is the Research and Innovation Sequencing Platform here in the University of KwaZulu-Natal School of Lab Medicine. He is also a research associate with CAPRISA. And for the most part of 2000, Richard has devoted his work almost fully on COVID activities, including clinical work, training, outbreak investigations, and research. So thank you very much for availing yourself for this discussion. We hope to have a lively discussion. You can just say your hellos, Richard.
Thank you, Kanye, and like Tulio, it's great to be here and great to have an opportunity to discuss a little bit more and kind of an open discussion.
Thank you very much, Richard. So we obviously, there was a gap in having this conversation about this virus that has visited upon the world and is obviously changing its forms and appearances on us and wreaking havoc in its path. Maybe we can just unpack it a little bit so that people who are joining us for this webinar, I'm aware that we have members of staff joining us. We have members of the alumni, and we also have members of the press. So you're welcome to ask questions at the end of the session. If we have time in between, we'll try and take live questions if time allows, but we only have an hour to discuss something that people spend a lifetime studying. So we hope we can do justice to it in just an hour. So maybe just as an opening to our discussion, we should just try and position ourselves and explain probably where we are as the world as far as the global pandemic is. Where are we and where did we start? You want to start that maybe, Tulio?
Yes. So let me start by the beginning. Would you mind if I share very quick my screen? Yeah.
Please do.
I don't know if you guys can see my screen, but that's a picture of all of us, including Kanye and Nathan, and that was in February 22, 2020. Yeah? That's for how long we have been preparing for the work that's happening now. Yeah. So it was very interesting just to start that, as you notice, this virus to appear in Asia and move to Europe. Yeah, we decided to take very serious, and here at the university, also with the health department and the NHLS, and we start preparing from then. Yeah, and if I can tell you, that probably was the main reason why we could do such a high level science. It is just the very hard work and preparation that we put that for almost a year. Yeah. And it has been a very busy year. Yeah. But I think that it is great to see, at least from us, yeah, that this preparation may have paid off, at least scientifically. We still end up with a terrible pandemic, which is still devastating and in the life of many people. But it was fantastic to see that had happened. Yeah. So I just thought that would be nice to start by discussing how we prepare, but also as a university, as we came really together. Yeah. And it was exciting coming together, not only within the university, but also involved the Department of Health, the Albert Lutuli, the King Edward, yeah, and how Caprisa, Ari, and many different scientists that we just unite to try to understand this virus almost in real time in South Africa. Yeah. So I just thought just to start with these early remarks that almost a year ago, it feels like 10 years, yeah, but it was only a year ago.
Yes, thank you very much, Tulio, for reminding us that. So let's take it to COVID-19, the state of the, yesterday we had the State of the Nation address. Can we perhaps present the state of where we are in the pandemic? Where are we with COVID-19? Where did we start? Are we seeing a downward trend? Are we plateauing? Are things getting better? Or are things, is there a light at the end of this tunnel? Richard.
Well, I mean, I think like you said, we've kind of entered a new phase of the epidemic, isn't it? With the emergence of these variants in different parts of the world, and at the time that we're starting to kind of try and rapidly roll out the vaccines, and essentially where we are now is where we've been from the beginning, but in a kind of race against the virus. And unfortunately, for most of this first year, the virus has been well ahead of us in that race, and it's really been a devastating epidemic. As Tulio said, we've not seen anything like this from a respiratory virus for over 100 years, and we know now we've had over 100 million cases globally, but we know that that's a significant underestimate of the true number of infections. We've had almost 2.5 million deaths globally, but again, that's probably an undercount of the true number of deaths. And I think that we see now there are still a small number of countries that have managed to control the epidemic and are managing to kind of live relatively normal lives. So we see pictures from Australia and New Zealand of rugby stadiums full of people in restaurants. They're going about their normal lives because they managed to control the virus from the beginning. And also, obviously, a few countries in Southeast Asia and things. But the vast majority of countries in the world have really struggled to control this virus. And that's certainly the case here in South Africa. And I think, like I say, what we're seeing now with these variants, some of them are causing us even greater challenges because of the changes that you described in the virus. They're making it harder for us to control the virus, and so we need to step up and really up our game if we're going to get ahead of the virus.
Thank you very much for that introduction and for basically painting a picture of where we are in the state of the pandemic. So you've also obviously made an introduction of the variants that are the topic of discussion today. Perhaps let us try and unpack a bit of terminology and try and explain what is a virus, when does it become a variant? We hear people talking of strains, we hear people talking of lineages. It may be confusing. Can we perhaps simplify the terminology and the taxonomy? Please, Tulio.
So I can try and do, can you? Because Tulio gets upset by this, that we use all this confusing terminology. So I can try. I mean, I think in terms of a virus, we know that a virus is something that infects different animals, but can infect humans and requires our cells to replicate. So it has to get into our cells to make copies of itself and to remain alive. When we talk about, one of the terms we need to understand is mutation, because that's key to everything that stems from that. And when we talk about a mutation, we're talking about a change in the genetic code. So in this virus, we're talking about a change in the RNA. And when that happens, usually it doesn't change the amino acid or the protein of the virus. So it doesn't make any difference to the virus, doesn't change its behavior or its structure. But sometimes that mutation changes the amino acid and changes one of the virus proteins. And when we talk about a variant, what we're talking about is a distinct virus with a certain pattern of mutations. And so in terms of the 501YV2, that was noted as a variant because it had a different combination of mutations than what we were seeing spreading in South Africa beforehand. The other term that's used is this term lineage, and really it's very similar, but lineage, if you just think of a family tree, when Tulio and the team are doing the analysis of the viruses, to compare all the viruses in South Africa and around the world, they kind of create a family tree of the viruses. And we can think about a lineage as just one branch of that tree. So the viruses are all similar on that branch, but sometimes they have slightly different mutations because they're changing on that branch. So that's my attempt at explaining the terminology, and Tulio can tell me what I got wrong there. But and the other term that's sometimes used, sorry, is strain. And again, yes, that's confusing. But generally, when we talk about a strain, we're talking about a variant that has got some additional properties, or it's got some change in its properties, whether that's that it's able to transmit more efficiently, so it's more transmissible, or it may cause more severe disease, but there's something about that virus that has changed, and then some people use this term strain. So they're all loosely defined terms, but that's a kind of explanation of what we mean when we're talking about these things.
I think that's a lovely summary, Richard. I don't know if Tulio has anything to add to that, but I probably want to pose another question that we should probe a bit more. So we're hearing different variants are popping up on all parts of the world. It's obviously not a uniquely South African issue. Do we perhaps know what's driving the emergence of these variants, and is there an end in sight, or should we just accept that we will always be living with mutating viruses and variants will be the order of the day? Tulio, I will pose that one to you. What is driving this?
So the reality is that we don't really know what's driving that. At the moment, there is two main hypotheses. The first one, it is that these variants with large number of mutations, because the difference between the three main variants that were discovered both in South Africa, in the UK, and initially in Japan, but was linked back to Brazil, is that they have many more mutations than the normal lineages that was circulating. So one thing, the first strong hypothesis of that this is developed in immune-compromised individuals. What it means, it would be individuals, for example, patients in long-term cancer therapy, that the immune system is low and couldn't attack very well the virus, and the virus would develop these mutations. Why this hypothesis, this very strong hypothesis exists? Because they had two of these patients that they have followed over time, and they saw the exactly number of mutations appearing. One of them was in the US, in the Boston area, and was described in a New England Journal of Medicine, that had both the mutations. This means the 501Y, that's the one that's common between all these three variants in the world. But it also had the mutation, the 484K, which is the one that we believe that affects the decrease on neutralization. And you may have seen quite strong on the news the potential effects on vaccines and also reinfection. And another one was another colleague of us that worked at ARI, Ravi Gupta. He had described that in Cambridge, in the UK, another patient that was immune compromised and developed these mutations. In the case, that one developed also the very similar mutations as the B117. That's the variant that have been described in the UK. However, also in the last few weeks, it came another strong hypothesis, plausible explanation, that these variants, they end up emerging in places that were severely affected by the first wave. For example, in Manaus, up to, they estimate, close to 75% attack rate. This means that when they did a serology, they found 75% of the people that have antibodies against the virus in the first wave. And also here in South Africa, in the Nelson Mandela Bay, when you look to the especially number of deaths there, they estimate that was the mostly affected area of the country. And then in the same also in London, in the UK. So it is emerging a second hypothesis that places that had a lot of infections from the first wave may have a lot of individuals that have decreasing levels of antibodies. And when this new virus come, it is not enough to completely neutralize and would become an effect of helping to select escape antibodies mutations. If we're gonna know which of them are the right hypothesis, I don't know. Sometimes it's a little bit like and you may see it this week that a whole team of the WHO went to China to try to look for the origin of the virus. And sometimes that's a question that we may not be able to answer. But I don't know what you think, Hani and Richard, if you agree with me or not.
Yeah, I mean, I think in simple terms, I mean, this virus is evolving. It's kind of to be expected that as more and more people are infected around the world, this virus is evolving in ways to allow it to continue to spread. And I think that what we're seeing with these variants is that there's evidence that they have these additional properties, that some of them are increasingly transmissible, and also that they may be able to evade parts of the immune response. And these are understandable because these may allow the virus to continue spreading in populations where there have been quite a high number of infections. And that's a good warning to us and a good reminder about the risks of not controlling virus transmission while we roll out the vaccines. Because if we roll out the vaccines slowly and allow the virus to spread while we're doing that, then there's going to be that additional, what's called kind of selective pressure on the virus, that some people have been vaccinated, others haven't, and the virus is spreading amongst that population. And what we'll see there is the selection of more variants that have the capacity to evade that immune response from the vaccines. And so that's kind of the key understanding about why the vaccine rollout has to be quick, but it has to be in concert with controlling the virus transmission.
Thanks, Richard. So, and I think a point that's also coming strongly in your response is always to be on the lookout and doing proper surveillance to see if there is emergence of these variants and how widespread they are, and what effect they would have on any measures we put in for either for prevention or diagnosis, or even treatment, because we know as viruses mutate, sometimes it may affect how they respond to drugs that are put up for treating. Maybe we can just share what the South African experience is on how did we find these variants? Because for a long time, we bore the ugly label of having a South African variant, as if it was a problem to find it. We could have just lived in a state of ignorance and not know there's a variant circulating. But the fact that we had systems put in place to do proper surveillance, it was detected, and it was detected timeously before a vaccine was rolled out, and we obviously could know and learn if it would be effective against the vaccines that were already developed. Maybe I can just, as a passing point, try and draw out how in South Africa we've gone about putting up a surveillance program for genomic surveillance, specifically for SARS in this case, but obviously in the future it may be used for many other emerging pathogens and other infectious diseases, which we know are a problem and an emerging cause of infections worldwide. In South Africa, we have about 5 labs and centers, research centers that have partnered with diagnostic labs, mostly the NHLS, as well as some private labs which are part of this genomic network of surveillance. And the idea is to sequence a small proportion of the viruses that are detected from patients when they are confirmed. And in that way, we get to know how the virus is evolving, and if any of the changes as the virus evolves have an effect in either how the epidemic is going. We know now that 5012 was associated with high transmissibility, which made us see a very steep and a sharp peak rise in infections due to the high transmissibility. Other countries have reported that the UK specifically is reporting that there may be evidence that there is an element of a more severe disease. We do not have solid evidence to support that here in South Africa, but it drives the point that if you do not look, you never know. So it's very important, and I think the South African science community should be lauded for that foresight of putting that type of surveillance program so that we know what is going on. And then maybe the next level of questions we should dwell into is, now that we know there are variants, what does it mean? Are we still able to diagnose this virus now that it looks a little bit different? Is the evidence telling us that we're missing some infections because of these variants? Any takers, Richard or Tulio?
So you would be the best person to answer that, can you? But I mean, I think that the evidence we have at the moment is that certainly the PCR diagnostics and the rapid antigen tests that are being used more widely now are still working fine at detecting the 501YV2. We have seen with some of the other variants, particularly the 501YV1 that's spreading widely in the UK, that it does affect one of the PCR tests slightly. And that's actually been an advantage for them because it allows them to detect the variant through the PCR test and not require the sequencing. And that's actually enabled them to do a lot of the kind of epidemiological and clinical analyses that have given them a better understanding of the variant there. But the variant here is still detected fine, and obviously that's something we're keeping a lookout for as the virus may evolve further. And so it's important that we keep tracking that.
Yeah, I'll definitely echo that as well. What we are seeing on the diagnostic call phase is that it doesn't seem to be that we're missing any of the infections. It also helps that a lot of the diagnostic methods, you mentioned the PCR, do not just look for one target. So you look at the virus at different angles, and even if one has mutated such a way that it may not be picked up, but you have a lot of other targets that still allow a diagnosis to be made. So that's very important to reassure the public that the mutations do not mean that we're missing the diagnosis of infections. Then speaking on transmissibility, what does it mean and how do we interpret this with schools reopening? Probably we will get an announcement in the not so distant future of the level of restrictions easing. Should the message that's conveyed to the public on how they prevent transmission, should it change? It's a big talk right now in the US about wearing double masks. Is there evidence that supports double masking? How do we translate the high transmissibility of this virus to how we prevent infection, transmission, or acquisition? Tulio?
Wow, Kenny, you give like 5 questions in one. Let me try to answer the first one. Yeah. Is this virus or this variant more transmissible? Yeah, the answer yes. We know quite well that these variants, they are around like 50% more transmissible than the previous lineages circulating both in South Africa and in the UK. That's when these variants, the different one dominating. Yeah? And that's because the change of the protein that allow better binding affinity at the ACE2 receptor, that's the receptor to enter the body. So we do know that, yeah. We also know that in the beginning was framed that the South African variant was more dangerous because there are the mutations, and we highlight quite early to the international scientific community and to the press that all these variants, they are going up in the fitness peak. And then what it means that in the last few weeks, the variant that was discovered in the UK, it's adding many of the mutations that's found in South Africa, yeah, that also evade neutralization. So it's just going back to what Richard said, that the main objective now is to try to also control transmission as we roll out the vaccine. Otherwise, this virus will keep outsmarting us. Yeah. In relationship to the schools, it's always a very difficult question to answer. We know a few things. We know that children tend to be less infected, or if they're infected, they have lower viral loads, so they develop much lower level of disease. We also know that it's important to send children to school, but we also need to know that we need to avoid big gatherings and super spreading events. Yeah, so it's always a balancing act. Yeah, and so So it's always a very difficult question. Yeah, we know that a lot of the private schools are already open in South Africa with strong protocols, but of course they are much more better resourced and with much smaller classes, especially than the more public and the private schools. So it's a difficult question. Yeah, in the UK they have decided not to open school until the virus is controlled at a low level. But I think that we're going to be hearing more about that in the next few days. Yeah, I don't know if Richard wants to comment on that.
Yeah, I mean, just to add a little bit to that, I mean, when we talk about the increased transmissibility, what we're really saying is that we have some evidence that this virus spreads more efficiently through the population. So it's spreading more efficiently from person to person. One of the things we don't know for sure is the exact mechanics of that. So what is behind that? So Tulio mentioned the kind of molecular aspect, that this virus hooks on more strongly to the receptors on our cells. So we don't know if that transmissibility advantage is just driven by people being more susceptible to being infected. So because the virus can attach more strongly, I'm more susceptible to get infected if I'm breathing air that has the virus particles in it. Or does these changes also make someone more infectious? So does it increase the viral load in my nose or in my lungs, and do I emit more of the virus if I'm somebody who's infectious? And it may be, like many things with this virus, it may be a mix of these things. But I think that the key principles about how we prevent spread from person to person are the same. I think what you're seeing in the US with this double masking is, you'll remember that with the mask wearing, the principle for most of the time we've been doing that has not been that we're protecting ourselves, but it's that we're protecting others around us. So it's, if we are infectious, we're preventing ourselves from emitting the virus particles into the air around us. But I think what you're seeing in the US with some kind of laboratory evidence and things is they're thinking of better ways that we also protect ourselves from inhaling and the particles. And so the double masking is kind of strengthening that protection. And we need to look at that evidence and see whether that's something that we want to adopt here. The reality is many of us, unfortunately, are not very good with single masking. So, I mean, the first principle is to try and get better throughout our communities at the single masking, and then we can maybe start thinking about the double masking.
That's indeed true, and thank you for clarifying that, Richard. And then maybe moving right along in the interest of time, the obvious big elephant in the room is the issue of vaccines and what the variants mean for vaccines. After the recent press briefing that we had with the Minister of Health, there has been some concerns in the public, which maybe we can try and simplify in trying to explain if, firstly, is the evidence saying that these variants are not as susceptible to the vaccines as expected? Is that what the evidence is telling us? If so, do we still have a benefit from these vaccines, and where can they still help us? Maybe we can try and tackle that first. Just clarifying what the evidence is saying and what the evidence was first, what these studies were looking for. Was it before the variants came along? Was it after the variants? And now that we have variants, what's next?
Yeah, I can just start with what we know from the variants, especially from the news on the AstraZeneca vaccine, because we participated in that paper, so we know quite well the innings of that analysis. Yeah, so that was a phase 1B, phase 2A trial, which is a small trial, 2,000 people, with the main objective just to see the safety of the vaccine. Yeah, and in this trial, most of people are young. The average age was 30 years old, or the median age. Yeah. So what's happened in the trial is in the both arms, the placebo and the vaccine, no one developed disease. Okay, that's why it has been coming on the news that we don't have data on
severe disease.
On severe disease, yes.
Exactly. Severe disease, not just disease.
Thank you, Richard. It's wonderful to have Richard next to my office. Yeah, and so that's what you know. No one develops severe disease. Yeah, and so what we also know is that in both arms there was similar number of breakthrough infections. What it means, people got infected with the virus. 23 on the placebo and 19 on the vaccine. Yeah, anyway, the vaccine main objective is not to avoid infection, is to avoid development of severe disease. But this data we didn't have. And then when they calculate the difference between what they call efficacy, which the trial was not powered to do, it only gave an efficacy of 20% because the 19 versus the 23. And then what we did, we sequenced the whole virus. So from those like 39 or 42 were the new variant, yeah. And then both in the labs of Penny Moore and Alex Siegel here in the Nelson Mandela School of Medicine, they exposed the virus to the plasma of the people that were vaccinated, just to see if that neutralized. And the neutralization was lower, yeah, was quite low, the neutralization. So what we know that this variant, yeah, which has escape mutations from antibody neutralization, is slightly less neutralized by the antibodies that are produced from the vaccines. Yeah. But also what we know that another trial that was done in South Africa, the Johnson & Johnson, which was a much bigger trial, was powered to see the efficacy of on severe disease. Yeah, it gave a very high efficacy result, was 80–90% efficacious on people not developing severe disease. And this vaccine also has a big advantage because it's a 1-dose vaccine, whereas a lot of the other ones are 2-dose. You vaccinated and you come back. How many days, Richard? 28 days?
In the trial, 28 days, yeah.
28 days to get the second vaccine, and these results, to come with these results before the vaccine is completely rolled out, is very important because then you can understand much more and you can have a better selection of the vaccine. So despite it being kind of, in theory, bad news, it was fantastic that came that as quick as could come. And that in South Africa, we have run these trials because then otherwise we wouldn't know what's the effect of these variants. Yeah, another vaccine that's likely to be very efficacious. Yeah, it is the Pfizer vaccine. It's a different technology, mRNA, and give much higher level of antibodies, and they show also T-cell response. Yeah, the difficulty of the Pfizer is that have to be store at very cold temperatures. Yeah. So one of the good things, knowing that very early, it is that also these variants can be added in the vaccines to either a booster or a modification. Yeah. What's quite positive. Yeah. But one important thing why we are worried about the AstraZeneca, because this vaccine, it was a vaccine that came from a very noble cause. The University of Oxford decided not to hold any royalties, and that's why it could be produced at much cheaper price, yeah, in generic labs. So it's very important, and we still think and we hope that this vaccine is going to be efficacious to avoid severe disease. Yeah.
And I think that's a very important thing to communicate, the severity of disease and the efficacy on severity of disease, because that is what we're trying to achieve. Yes, Richard?
And maybe just worth just explaining to people some of the principles behind that. So as Tulio explained, so the main changes that we see in the variant are changing the shape of this spike protein. And what we've seen now with all the evidence that we're collating is that has an impact on the binding of many of the neutralizing antibodies. So essentially, these antibodies are supposed to bind to the spike protein and stop it from hooking onto the receptor on our cells. And that's what can prevent infection, as Tulio said. But the bit of the immune response that kind of mediates the severity of disease is more the T cell response. And what we know about the T cells is that they seem to target many different parts of the virus. So not just the spike protein, but other parts of the virus. And that's what kind of underlies our thinking that although this variant has lots of mutations, the T cell response recognizes so many different parts of the virus that it's less affected by the mutations. And that means, and that's kind of our thinking behind, although this may have a problem with not preventing infection and mild disease, it may still prevent severe infection. And it's just now a matter that it's when you've got these different vaccines that you really need the evidence for each one of them to understand that. And the reality is, I mean, the AstraZeneca trial has had a lot of criticism and things, but the reality is it was just not designed to look at severe disease. So it's not really fair to criticize the trial and the groups that ran the trial, because it did what it set out to do and answered the questions it set out to answer. And it shows that actually, like we all know, science is not easy. Science is difficult, and science we don't always get all the answers that we want from the research that we do, and we have to interpret things in light of what we learn from multiple different things, and that's what we're doing at the moment is trying to put lots of different pieces of evidence together to work out the best strategy to move forward.
Thank you, Richard, and that's a very important explanation. Obviously, the natural question that arises from that is a vaccine will always be a few steps behind the mutation of the virus, and viruses will always mutate. How then do we keep up and create vaccines that probably have a broader response and are not always tailor-made to a specific a virus in future, considering that what we have with influenza, is this the direction that coronavirus is taking? Or are we more likely to just settle with a vaccine that alters the severity of disease without necessarily preventing transmission of infection? What's the future most likely to be? And we just have 5 minutes to go into questions.
Yeah, I mean, it's a great question. I mean, I think what we're seeing is that most of the vaccine manufacturers are now already working on these modifications to deal with these variants that have emerged. But as you say, that's a kind of reactive thing. We're reacting to what we're seeing now. And by the time we've developed those modified vaccines, maybe the virus has evolved further if we've allowed it to. I think the other thing that it highlights is the need to develop different types of vaccine that, like you say, are not just targeting the spike protein, but are targeting different parts of the virus that maybe are less prone to these mutations that we've seen. I think that the spike protein was the obvious target for vaccines when we started learning about this virus, because that's what hooks on to our cells. It's what mediates the entry of the virus into our cells. So it was the right target. But now that we know that that's prone to multiple mutations, I think that there is a need to develop and evaluate kind of next generation of vaccines that target different parts, different proteins within the virus. And I think that's what you will see over the next few months is more vaccines being developed there.
Yeah, but just to add, and an interesting thing for people that's interested on the impact of large rollout of vaccines is to see what's happened, for example, in Israel. Yeah, they are the country most advanced. Yeah, and so far the output that they are getting there on severe disease was much better than they ever expected. Yeah, and so we are also seeing that in these vaccines, if you roll quick to a large number of population, not only decrease radically disease and hospitalization and death, but they also saw decrease on transmission. So it's quite exciting, and that's why a few countries are rolling so fast the vaccines. Yeah.
Thank you very much. I think in the last 5 minutes we have left on the webinar, we'll just open it out for some live questions. I'll go through some of the questions that we already have in our chat group and see if any of those have not been addressed during the discussion. There's a question here from Bonginkosi Chiliza asking, what will it take for us as UKZN or KZN to develop our own vaccine manufacturing capacity? Any takers?
Great question. I mean, it would take a lot. It would take a lot of commitment, a lot of financial investment. But I think that we've seen here, and I'm sure this is where the question's coming from. We've seen the problem with the current system, where a lot of vaccine development and manufacture is in high-income countries, and what we're seeing is this real inequity in the distribution of vaccines there, and like with many things, Africa as a continent is at the back of the queue for the vaccines, and really that highlights how unacceptable that is when you have a global pandemic like this. And so there certainly could be much more thought given to how Africa as a continent, South Africa, develops the capacity to do the science, to do the development and the manufacturing of vaccines on the continent. And if UKZN was to play a leading part in that, that would be absolutely fantastic. I mean, but it would need to be, it would require a huge amount of political commitment and investment, and resources, but why not? I mean, this highlights how important that kind of thing is.
I have another question here from Bumi asking if the variants are linked to populations that have high levels of infection in the first wave. Why are we not seeing more variants coming out of the US? I think the logical question there is probably the level of surveillance that they're doing is different. You find when you look, I'm not sure if there is intensive and active surveillance to track and monitor variants as they emerge. That probably would be the answer to that.
Yeah, and I can also answer that in the last few weeks, they detected 2 potential variants, what they call now under investigation. One is in California that has some mutations in the spike. The other one was in Ohio, yeah. And what they have seen is that the variant that emerged in the UK is beginning to take also the US by storm. Yeah, what it means is really growing very fast in prevalence, especially in California and Florida, because as you imagine, a lot of flight connections between the 2 countries. Yeah.
There's another question here saying South Africa did not have a large epidemic in the first wave, yet we describe the 501V2 as the prevalent variant. Does this not contradict one of the hypotheses? I think here it's a timing question. Perhaps, Thule, you can tell us when exactly this 501 emerged and why was it not behind the first wave? Thanks.
Okay, I will do, but the first thing is that South Africa had a very big first wave. I don't think people realize how big it was. Yeah, it was more than 50% of all the infections in the African continent. Yeah. And during that time, especially in some of the more deprived areas of the country, surveillance and testing was not done at the level that was needed. And we can see that, for example, that the WHO, they get very worried when the positivity rate of testing in a country passed the 5% mark. In the UK, when past 1% mark, they put extreme lockdown. In South Africa, we celebrate when it's at 10% or 15%. So that's the first misconception, and especially where you think that this variant had emerged. Doesn't mean that the variants originated from there, but emerged with the second wave, the Eastern Capes. Some of the data there is quite extreme. I don't know if Richard wanna add on the mortality, what they estimate on the size of the epidemic in the Eastern Cape in the first wave.
Yeah, I mean, it's just a reminder that what we see with the reported number of cases and the deaths is a significant undercount of the true number of infections in the community and the true number of deaths, and we know that because we have an excellent mortality surveillance system in South Africa, one of the best in the world, and that allows you to track the total number of deaths, and you hear that talked about as excess deaths. So you can look at what you expect the number of deaths to be based on the past few years, and then what the true number of deaths is. And we're now sitting at almost 140,000 excess deaths in South Africa after the second wave. And when you convert that to a population level, and when you think about our young population here in South Africa, that really is one of the worst levels anywhere in the world. And when you focus down on specific areas, like Tulio says, of Eastern Cape, Nelson Mandela Bay, the rates are really exceptionally high. And so unfortunately, we have been kind of, there's been misunderstanding throughout the epidemic a little bit that we've been not badly hit by this virus and lightly affected, and the reality is, and many of us unfortunately now know that from our own personal experience in our own families, in our communities, we've been extremely badly affected by this pandemic.
Thank you. Maybe as a last discussion point, I see we have our DVC who's raised her hand. I don't know if we can allow her to ask a question. Professor Nama?
Sorry, I didn't mean to raise a hand. Thank you so much. I didn't mean to raise a hand. I was trying to type the question, and the question is basically asking, as a follow-up to what Prof. Chilisa has asked, whether we have the skills or do we have to look at importing skills for development of vaccines? Thanks.
I can start asking that. Yeah. One thing that South Africa has been showing around the world is that we have fantastic science in the country, and we are really being praised. Today is going to come a very nice piece on the NIH, a blog from the director of the NIH, Francis Collins, that he will highlight how level science has been done in South Africa. That's a combination from both people in South Africa, but also large investment from international sources. Yeah. One thing that we see in all the top academic research institutes in the world is this big diversity. Yeah? And diversity has been shown to increase scientific level just because people think by different ways and they can come together with the answer. Yeah. and it's very common. For example, you go to Stanford or Oxford, yeah, some departments, 60% of people will not be from the country, just because all this diversity really increases the level of scientific question. Yeah, so I really hope that by we portraying internationally the very high level of the science that has been done in South Africa, that will increase the amount of investment both internally from our government, yeah, but also that we can attract more international funding to really make South Africa one of the top scientific destinations for kind of pathogen research in the world. Yeah.
Yeah, and just to add that it's important to remember that although kind of Tulio and I appear on these webinars and on these TV and things, we're not the ones doing the actual work on a lot of this science. The people behind us that are doing the actual work are the PhD students, the postdoctoral students as part of CRISP, as part of UKZN. The person who took the new variant and worked out how to grow it in the laboratory, to grow the live virus. Sandile Eli, a PhD student again in Alex Sigal's lab at ARI. These are the young scientists, and this pandemic has kind of created an opportunity for them to see how their science can be so impactful in such a short space of time, that you're doing the science in real time. It's giving you answers that have global significance. And I think that we have to really take this opportunity to show how strong that is here at UKZN, here in South Africa, and to encourage the next generation of scientists to do science and to see how that can really contribute to society.
Yeah, and then just to add one thing that you may not know, yeah, is that the first author and the second author of the Network for Genomic Surveillance in South Africa is Kanye and Koleka. Yeah, and the reason why most of people don't know is because the NHLS has a moratorium that they cannot talk to the media. I was discussing with Koleka to, please guys, you have to talk with your CEO, because if it was not Kanye and Koleka with really strong support from the NHLS, we wouldn't have such a successful network. Yeah.
What a note to leave this discussion on, Tulio. Thank you very much. I think we've come to the end of our session. We've just gone over 10 o'clock, and I hope this was helpful in trying to open up the discussion and unpack what this variant means to the epidemic, what it means to our efforts to try and prevent it, and obviously what it means to treatment. I see some of the questions we're asking on ivermectin. I understand there was another webinar yesterday discussing specifically the topic of ivermectin and other treatment options, and I'm afraid we could not go into details on that today. So with that, I'll just allow our speakers, our panelists, to just have their parting shots, and we will bid you goodbye.
Thanks very much, and thanks for moderating a great discussion, Kanye, and I hope that there may be an opportunity to do this at more regular intervals, because I think actually this chance to kind of have a dialogue is really helpful, that it's not just us presenting slides and things, but an actual dialogue, and I think that helps to kind of focus in on some key aspects. It's been great, and thank you very much.
Thank you very much, Richard. Tulio?
Yes, the same from me, Kenny. As you know, I'm gonna do my weekly visit to your lab in the next few hours. And so it's great to debate with you in person, but it's also fantastic to do that in this venue of UKZN. And thank you very much for the whole support from the university, to both CRISP, but also Department of Virology, and to allow us to give the tools to make such a high level science in South Africa. Thank you.
Thank you very much. And thank you very much to the university. Thanks to our DVC who graced us with her presence today. We really appreciate having these opportunities to have informative sessions and have science engagements that make science living and alive and explain it in simple terms so that we take each other along in discoveries and in putting South Africa forward. Thank you very much, and may we all have a lovely